dear students. Welcome to the next lecture of organic chemistry. In this lecture, we shall discuss about enzymes. What are enzymes? Enzymes are specialized class of proteins with high molecular masses. They are biological catalysts which catalyze various biochemical reactions in the cell. They are capable of acting independently of the cells. The enzymes which function inside the same cells which produce them are called endoenzymes or intracellular enzymes, while those enzymes which function outside the cells which produce them are called extracellular enzymes or exoenzymes. The molecules upon which the enzymes act are called substrates. Enzymes convert substrates into different molecules called products. And after the reaction, the enzyme is regenerated. The graph shows a comparison of the conversion of reactants into products in the presence of enzyme and in the absence of enzyme. In the absence of enzyme, if we consider the reaction, the free energy change with progress of the reaction, we can see that the activation energy is a higher value compared to the reaction in the presence of an enzyme. Thus, enzymes decrease the activation energy of the reaction and thus accelerates the conversion of the reactant into product. A few examples of enzyme catalyzed reactions are shown here. The enzyme invertase catalyzes the hydrolysis of sucrose, and the products obtained are one molecule of glucose and one molecule of fructose. Starch is converted to maltose by the enzyme diastase. Maltose is converted to two molecules of glucose in the presence of maltase enzyme. The decomposition of urea into ammonia and carbon dioxide is catalyzed by the enzyme urease. Next comes the important features of enzymes. Enzymes are highly specific for a particular substrate. They remain unchanged during the reaction. Enzymes are very efficient and they catalyze about 1 to 10,000 molecules of substrate per second. Enzymes speed up the attainment of equilibrium in biochemical reactions. The characteristic temperature at which an enzyme shows the maximum catalytic activity is called the optimum temperature of that enzyme. And each enzyme shows a maximum catalytic activity at a characteristic pH, and this pH is known as the optimum pH of that particular enzyme. The catalytic activity of most of the enzymes is enhanced by the attachment of a non-protein part called cofactor. The cofactor forms a bridge between the enzyme and the substrate and thus enhances the activity of the enzyme. The figure represents the attachment of a cofactor to the enzyme. For example, the enzyme alcohol dehydrogenase is activated by zinc 2 plus. Amylase enzyme is activated by Na plus, etc. This non-protein part which enhances the catalytic activity of the enzyme is known as the cofactor. The catalytic activity of an enzyme is decreased or completely destroyed by the presence of certain substances called inhibitors. An inhibitor functions by interacting with the active functional group on the enzyme and thereby inhibits the function of the enzyme. Heavy metal ions like silver, mercury, lead inhibits the activity of enzyme urease. The activity of succinic acid dehydrogenase is completely inhibited by the presence of malonic acid. Thus, the substances which decrease or completely destroy the catalytic activity of an enzyme are called inhibitors. Enzyme activators and inhibitors are collectively known as enzyme modifiers or enzyme regulators.